Hi, everyone. It's really great to be here. My name is Hannah Ledvina. I'm currently a postdoc in Aaron Whiteley lab at the University of Colorado in Boulder. Before starting here, I did my graduate work in Joseph Mougeot's lab at the University of Washington, studying the type 6 secretion system. But today I'm really excited to share with you my work looking at how functional amyloid proteins confer defense against predatory bacteria. So every single day, all of us are bombarded with many different pathogens, both bacterial and viral and fungal. And in order to counter these, we have evolved sophisticated immune pathways that specifically detect and respond to these different pathogens in order to elicit the proper immune response. We have many different pathways here. I've just illustrated two different ones and demonstrated how they can elicit a different immune response dependent on the pathogen. Bacteria, just like humans out in nature, are faced with many different pathogens. The one we're most commonly thinking about are bacteriophages or phages, which are the viruses that infect bacteria. And just like humans, bacteria also have sophisticated immune systems that are capable of recognizing these infections and eliciting the proper response. Two that you may be familiar with are restriction modification pathways and the CRISPR-Cas pathway. Both of these are kind of historically well-known immune pathways within bacteria and have been adapted to be really important biotech tools that many of us use within the lab. But over the last few years, this field has really exploded, and recently we have found that bacteria encode over 100 different immune pathways that can respond to phage infection through different means. And what's been particularly exciting about this is we found that many of these are evolutionarily related to the human immune pathway, and therefore we can learn a lot about how the immune pathway works in humans by studying these pathways within bacteria. For example, in my early postdoc work, I studied one of these pathways where we found that it is regulated by these E1, E2 proteins, which also regulate immune pathways within humans. But today I want to tell you about a different threat that bacteria face. So here I'm showing you two different cells. On the left, we have E. coli, which you're probably pretty familiar with. And then here on the right, I have a very small bacteria called Delavibrio bacteriovorus, or I'm just going to call it Delo for short. And if we just look at these two bacteria, here we're looking at some transmission electromicrograph images. They both look pretty harmless. We see one is much smaller than the other, but nothing too dramatic is happening. But when these two meet, something quite crazy happens. Delo is able to smash its way into the E. coli cell and grow within this compartment. So here now we're looking at a new image where you can see the E. coli has been overtaken by this smaller bacteria and has become a home for them to grow and divide and eat. And so, as you can imagine, this is quite devastating for the E. coli host. And so something I wanted to ask is, can E. coli stop this? Do bacteria possess any immune pathways dedicated to stopping this new type of threat? So not phage, but predatory bacteria. And so I'm not the first one to ask this question. Many people have studied Delo before, but they have often found that there's absolutely no defense against Delo. And this seemed really interesting to me, and I wanted to follow this up and better understand what was going on. And so most work studying Delo and how E. coli might block it use what we call lab-adapted E. coli. So here again, I'm showing you an image. This is E. coli on a plate. It's probably what you're used to seeing when you think about E. coli, just kind of a normal-looking colony. But out in the wild, E. coli is actually really diverse and takes on many different morphologies and colony types depending on the nutrient and the environment that it is grown in. And so in order to try to uncover a new immune pathway and figure out how E. coli can stop predatory bacteria, I wanted to use the diversity within the E. coli species. And so what I did is I got this collection that is called the E-core collection for the E. coli reference collection. And this is 72 strains of E. coli collected from all over the world from different environments. And importantly, they're all genetically diverse and represent the genetic diversity of the entire E. coli species. And so what I did is I took each of these different E. coli strains and I infected them with Dello. And what I looked for are plaques or holes in a lawn of E. coli where the Dello has been able to successfully infect and kill that strain. So here I'm showing an image with the red arrows indicating the plaques. So this is somewhere where the Dello was able to successfully infect. And so I can use this as a proxy to determine how resistant or susceptible an E. coli strain is. And so that's exactly what I did with this E-core collection. And so that's the data we're going to look at here. On the y-axis, what I'm plotting is the PFU per mil, or the plaque forming units. So how many of those plaques did I see when I infected the different E. coli strains I'm going to show along on the x-axis? Here, first I'm showing a strain called MG1655. This is our normal lab-adapted strain, so one we know that Dello is able to infect. And here you can see we have about 10 to the 7th PFU. So then I started looking throughout the E-core collection, and at first we found that most of the strains are looking just like our control, where we're seeing many plaques being formed. But every now and again, we would see a strain like this one down here, where we're down at this dotted line, which is our limit of detection. So I was unable to visualize any plaques at all on these strains, showing that they're very resistant. And indeed, when we looked across the entire collection, approximately a third of these strains had this phenotype where we saw absolutely no plaques, showing they are highly resistant to Dello infection. And so in order to figure out what was going on, I'm going to focus on this one here in purple, which is called E-core 14. And we're going to use this strain to try to determine why or how these E. coli strains are resisting Dello infection. What I did is I took E-core 14 and I performed transposon mutagenesis to create a library of roughly 7,000 individual mutants. We then took each of these mutants and infected them individually with Dello. And so now we're looking for a strain that can successfully infect because E-core 14 is normally resistant. And so here on the right, I'm showing an example plate where hopefully you can see this well here on the top is much more clear. This indicates that Dello was able to successfully get in and infect and kill that E. coli strain. And so then we can use PCR to map where that transposon inserted to determine where in the genome this mutation occurred, and therefore what are the genetic determinants of defense. And so to cut a long story short, I'm just going to jump to the end of what we figured out with this screen. And turns out defense all comes down to this extracellular structure called curli. So curli are extracellular functional amyloid proteins. Here on the left, I'm showing you an image of E. coli, and these little hair-like protrusions sticking off the top, these are the curli. Here on the right, I have a cartoon depiction of what these look like. And the main feature I just want to highlight is that they have this amyloid fold. These are highly durable proteins that are resistant to most breakdowns. You have to use things like formic acid in order to degrade them. And the word amyloid might sound familiar because this is the same protein fold that is the fold that causes things like causes amyloids in your brain, causing diseases such as Alzheimer's and Parkinson's disease. And so it was really exciting to find amyloids within bacteria that may be playing an immune role. And so what I didn't tell you is this image here on the left is of a strain that is susceptible to infection. So yes, it's making a little bit of curli, but it's just kind of this little dash off to the side. And so what we wanted to do is now take our E-core 14, our strain that is resistant, and see what it looks like. And here it is. And what I hope you can appreciate is that this strain is just completely coated in these fibers that are taking up the entire surface. We can barely even see the E. coli in there. It's just totally coated in these fibers. We know that this is the curli because when we make a mutation to genetically delete these, we no longer are seeing these fibers on the surface. And so what we can say is that our resistant strain is making significantly more curli. And so I've hinted that this is what's underlying defense, but let me actually show you the data to support that. Here, what we're looking at again is a PFU measurement. And again, with E-core14, this is our one that is resistant, so we were unable to see any plaques on this strain. But now when we delete the curli genes, we see that the strain becomes very susceptible, and we have levels up to where we would expect with our susceptible control. And indeed, if we now take our susceptible strain, MG1655, which normally has lots of plaques, and we overexpress curli, we are now able to reduce the ability of Delo to infect this strain. So curli are both necessary and sufficient for defense against Delo. But how exactly is this working? Let me walk you through the Delo life cycle. So out in the wild, Delo is just going to be swimming around and looking for a prey cell. So here I have Delo in green and an E. coli here in white. When they encounter each other, the Delo is going to attach to the surface of the cell, burrow its way into this cell where it will grow, divide, eventually lyse that host cell to escape and go on in order to infect additional prey cells. And so when we think about where curli is localized on the cell, on the membrane, kind of the natural hypothesis would be that curli is limiting attachment and entry by forming some sort of barrier along the surface. And indeed, if we use EM again to look at the cells during infection, that's exactly what we see. So here on the left, again, we have our wild type E-core14, and on the right, we have a mutant that is unable to make curli. Both of these have then been infected with Delo, which are these much smaller bacteria in the images. And what I hope you can appreciate is that for the wild type, there's this barrier of fibers blocking the ability of the smaller bacteria to access the much larger E. coli cell, and therefore it is unable to initiate that infection process by getting into the host. And so to kind of make this more simple, here's a model, the idea being a susceptible prey. The Delo is able to get to the membrane and burrow its way in. However, when a strain is making a lot of curli, it forms kind of a suit of armor, and so the Delo blocked off and is unable to enter into the cell. And so this was really exciting for many reasons. This is one of the first examples of an immune pathway that is acting outside of the cell. Typically, we think of immune responses happening once infection happens within. And so this was really a new paradigm for how immune systems within bacteria can function. And I like to think of it as kind of like the skin of a bacteria. You know, the skin is our first defense against pathogens. Curli might provide a similar defense against incoming pathogens. And so what I wanted to ask is, how common is this? Do other bacteria do this as well? And so the first thing I did is I went back to all of those different E. coli strains I had that were resistant to Delo. And indeed, what I found is for all of these, here I've just depicted a few of them, for our wild type, which are the pluses here, we see that we're getting no plaques, but for all of them, when I mutate curli, so they're unable to make this fiber anymore, we see they become highly susceptible to infection. So using this barrier is a common mechanism within E. coli to defend against predatory bacteria. But then we wanted to step back and look even further outside of E. coli and see if other bacteria are doing this as well. So here I'm showing you some results from some bioinformatic analysis we did. There's a lot here, but basically what I just want you to focus on is where we're seeing green. This is where we're seeing these functional amyloids, these amyloid-like proteins being made by bacteria. And all I really want you to appreciate is that we're seeing that they're in a lot of different bacteria. Many different bacteria are making them and making different components of them. And excitingly, our analysis also uncovered a different type of amyloid that's related, that are made by the Pseudomonas species. So Pseudomonas is a different bacterial species, and they make an amyloid called the FAP amyloids. And so what what we wanted to next look at is whether these are also able to defend against Delo. And indeed, what we found when we expressed these in our E. coli strain, as I've shown before in our gray bar, here we have our wild type where we're seeing lots of plaques. If we now overexpress this new amyloid, we see we have lowered PFU, and therefore these are able to defend against Delo just like the curli. And so we also wanted to look within Pseudomonas itself, and we saw something very similar to what we saw with E. coli, where if we look at many different strains of Pseudomonas here across the x-axis, what we see is many of them are resistant and some of them are susceptible, just like with E. coli. And similar with the curli observation, if we overexpress this FAP amyloid, we also are seeing reduced plaques. So what we can say is both these curli amyloids as well as the FAP amyloids are able to defend against predatory bacteria and therefore are a new branch of the bacterial immune system. And this was very exciting to us for many reasons. One of them is that bacteria are actually reported to make this whole array of different functional amyloids, many of which we really don't understand how they work. And so potentially many of them are actually involved in the bacterial immune system. And so we're excited to start uncovering the potential function of these. This is also particularly exciting for us because these bacterial amyloids have been linked to neurodegeneration within humans. And so potentially studying these bacterial immune pathways will allow us to develop tools and systems to combat neurodegeneration. But then looking forward, one thing that I'm really excited to pursue is the idea of studying bacterial immune systems against diverse threats. So I talked briefly at the beginning about all this work we've done investigating how bacteria defend themselves against phages, and I've now shown you how we can uncover interesting pathways by studying predatory bacteria as well. So here on the slide, what I'm showing you is many different species of predatory bacteria, most of which have not been studied at all. So the predatory bacteria are highlighted with the red arrow and their host is in black. And what this is meant to indicate is just the diverse life cycles and lifestyles of these predatory bacteria. Many of them will invade into their host, but several of them will stay extracellular. And so by trying to understand different ways that bacteria can defend against this, we can expand our understanding of the bacterial immune system. I've already started this by looking at one particular predatory bacteria. It's called Myxococcus xanthus. It's this one in the upper right here. And what we found is curli not only defends against Delo, but is also able to defend against this highly divergent predatory bacteria called Myxococcus. So here on the top, I have a bacteria that is unable to make curli, and on the bottom one that does. And what I hope you can see is the one that's unable to make curli kind of has this zone of clearance in the middle. This is where the Myxococcus was able to come in and eat those bacteria and kill them. But if the bacteria are making curli, it seems to be able to block this. And so I'm excited to continue studying these predatory bacteria to better understand this interplay between the two species. And so from this, what we're hoping to uncover is insight into unappreciated aspects of the bacterial immune system, which has really just been focused on phages in the past. Additionally, we're hoping to inform the design of novel biomedical tools. As I indicated earlier, we've talked about CRISPR and RM system and all the amazing things that those have been able to do. Potentially, some of these new systems will be the next system that will be able to make advances within our field. And finally, I'm excited to continue to uncover cool and interesting new science by studying these underappreciated organisms. And with that, I would like to thank my lab, first and foremost, my mentor, Aaron Whiteley, who has allowed me to take on this kind of interesting new direction in the lab of studying predatory bacteria. All the different members of the Whiteley lab, but in particular, the different students I've had the opportunity to train. I've worked with 4 amazing students, Ryan, Rico, Lexi, and Layla, all of whom made significant contributions to this work. And finally, I would like to thank all of our different funding sources, in particular, my funding through the Jane Coffin Childs Memorial Fund, without whom I would have been unable to do this work. And I'd also like to thank all of you for taking the time to listen to my work. And yes, with that, thank you so much.